Hey everyone, welcome to The Explainer. Today, we are embarking on a truly fascinating journey. Think of it as a micro-safari. We're going to venture deep into the hidden, unseen ecosystems that are thriving right under our noses, perhaps even right now in your very own kitchen. I am so excited to guide you through this.
But it begins with a pretty compelling question. How do we actually see the invisible? I mean, right now, the air around you, your desk, the pen in your hand, all of these surfaces are teeming with complex life, completely unseen by our standard senses. To explore this hidden wilderness, we're going to need the right scientific gear. So here is our expedition map for today. 1. The invisible world. 2. Gearing up for observation. 3. Microbes in coconut water. 4. Fungi on spoiled bread. And finally, 5. Accelerating microbial growth.
All right, part 1, the invisible world. Let's define our hidden habitat.
Basically, our targets are microorganisms. And whether they're unicellular, meaning they operate as just a single unit, or they're complex multicellular structures, the defining characteristic is that they operate way below the threshold of human sight. You simply cannot observe them clearly with the naked eye.
Moving on to part 2, gearing up for observation, the tools of the trade.
To actually breach that threshold of human sight, we rely on the compound light microscope. But successfully spotting our targets requires some rigorous, safe lab prep. First off, you absolutely must use a clean container so outside contaminants don't skew your results. Next, just place a single drop of your specimen right on the glass slide. Now, step 3 is crucial. When you apply that thin glass cover slip, you have to drop it at a slant. Doing that pushes the air out and prevents trapped bubbles from blocking your view. And finally, always start your observation on low power so you can capture a wide landscape before zooming in.
Okay, part 3, microbes in coconut water. This is our first safari destination.
To explore this first habitat, we set up a quick procedure where we leave a small amount of king coconut water exposed to the air for 3 days. When we take a drop of that water and view it under the low power of our microscope, we get a total aha moment. Notice those distinct circular-shaped cells? That is yeast. It's a type of unicellular fungus. And here's the crazy part. It wasn't in the water originally, it fell right out of the air. The coconut water simply acted as a nutrient-rich food source, allowing the invisible airborne yeast to rapidly multiply until there were enough of them for us to catch under our lens.
Section 4, fungi on spoiled bread. Get ready for a fuzzy encounter.
For this everyday encounter, our procedure is simple. We spray a slice of bread with a little water and just leave it sitting out for 3 days. Looking at the macro view, the food spoilage is super obvious, right? The microbial activity has totally altered the bread's color and texture, producing these gross, fuzzy patches. It ruins the nutritional value and makes it completely unfit for consumption. But what exactly is creating all that fuzz? Well, when we prep a slide of that fuzzy substance and look through our microscope, we reveal this totally alien landscape. This is a specific fungal variety known as mucor. If you look closely, you can see these really thin, white fiber-like threads, and they're supporting those dark, round structures at the top. This entire multicellular network is the culprit, actively digesting your sandwich.
This side-by-side directly connects the macro effect to the micro cause. We experience the spoiled bread with our naked eye, but it's the invisible mucor fungi working relentlessly behind the scenes that actually drives the whole process.
And that brings us to part 5, accelerating microbial growth, the rules of the microscopic jungle.
So, why exactly did the bread spoil and the coconut water grow yeast? Well, microorganisms, like any living creature, require really specific conditions to thrive. Warm room temperatures, we're talking between 25 and 30 degrees Celsius, combined with moisture, are the absolute ultimate accelerants. In a moist, warm environment, microbes multiply rapidly, and their enzymes break down the food via fermentation or putrefaction. Conversely, refrigeration acts basically as a pause button by artificially lowering those exact 2 factors, temperature and moisture. That right there is the fundamental secret to food preservation.
As we wrap up our safari today, I want you to remember this really powerful quote from the source text. Microorganisms are found in every habitat on the Earth. Think about that. They don't just exist in our little lab experiments on bread and coconut water.
They thrive in the atmosphere, in the water, deep in the soil, and living in and on plants and animals. They even survive in the most extreme, hostile environments you can imagine. Icy glaciers, boiling hot springs, barren deserts, and high-pressure deep-sea trenches.
Which brings us to our final thought for this explainer. If this massive, biologically diverse world of bacteria, fungi, algae, and protozoa is thriving in the air, the soil, and literally right inside our kitchens, what else is hiding right under your nose, waiting to be discovered? I highly encourage you to keep questioning the invisible world all around you. Thanks for joining me on this micro safari, and I'll catch you in the next explainer.